3 p.m. in the Netherlands, all around the world today for a very important topic that is changing people's lives. Very welcome to the Epic Online Technology Meeting on Photonics for Pharmaceuticals. Today is a key day for everyone. I just want everybody to remember that this meeting is all about creating business. This meeting is all for you to realize that today we can make business happen. Water. Relax. The season 2 of the Epic Online Technology Meetings is coming to an end. On Monday, November 30th at 3 p.m. Central European Time, on the episode Lucky 13, we reach out to one of the world's biggest and most important markets, pharmaceuticals. The average cost to research and develop each successful drug is about 2.6 billion euros. And in normal times, it takes at least 10 years for a new medicine to complete the journey from initial discovery to the marketplace. Photonics already plays a crucial role in the mass production of many drugs. Cameras ensure that exactly the right amount of active ingredients are packaged in exactly the same way, because tiny mistakes could cost millions. But how will the processes change if countries adopt personalized medicine? The growing field of precision medicine separates people into different groups. Medical decisions, practices, interventions, and drugs are tailored to the individual patient based on their predicted response or risk of disease. So, will 3D printing technologies allow for affordable personalized medicine? How will lasers be involved in quality control? And what role could AI and sensors play in reducing the length of clinical trials? Expect answers on Monday, November 30th, when Epic is organizing a meeting of the clinical minds. We are involving end users from the pharma world to explain the challenges ahead for the next 5 years. I am looking for input from Roche, Bayer, AstraZeneca, or Merck, and will also bring photonic companies like Lytid, HiSpecs, Specking, Xenics, and Thermo Fisher to explain their role in creating the safest medicines in the shortest possible time. It is time to adapt the remedy to fit the disease. So on the next 2 hours of your life, we are going to find business in the pharmaceutical market, and you will hear from Roche, and then we'll try to compare with the current state of the art and see how we can help the key companies in the sector. I want to say that these are fantastic online technology meetings. We keep receiving amazing feedback from all of you. I think you know by now that we are a fantastic family of already more than 600 members, and we became the largest association of photonics in the world, but that really doesn't matter. What really matters is that we keep bringing business to the industry and we keep getting your feedback. We only have 4 online technology meetings scheduled until Christmas, and we're going to finish with food and beverage. Mark that one in your calendar because it's going to look excellent. But we already last week announced the online technology meetings for Q1 2020, and it's true. 20 meetings in 3 months. We are basically going completely crazy, but we can do this. We are already actively scouting end users who are going to be the star of the show of these online technology meetings. You have all this information in the website. Tell us which events are most relevant for you and why, because we want to make sure that you appear in the right market, technology, and potential business opportunity for your company. Also, let me remind you that I'm here on behalf of a fantastic team. Elena and me are chairing today's meeting, but behind us we have an amazing group of people, technology experts, business experts, marketing experts, all of them there for you to help you with your business, in recruitment, in raising investment, and most important, in increasing your business. Today we are talking about the pharma industry, and this meeting would not have been possible within 2 groups of companies. First one, our media partners. Thank you very much, AZOnetwork and Medical Device Development, for supporting us and for communicating the way it deserves the opportunities for EPIC members in this market. And most important, thank you. Thank you very much. The two companies who are sponsoring today's meeting. Fisba, if you are looking for optics, if you are looking for optical solution and micro optics for the medical market, Fisba is the key company who can provide any beam shaping, any light shaping for any market. And Modulight, all the way from Tampere, Finland, semiconductor laser manufacturer. This company makes MVE growth to all the way to make devices that serve clinicians. So from the growth to the clinic, they cover the entire value chain. An example for the European industry. Once again, thank you, Fisba and Modulight, for sponsoring today's meeting. And thank you very much, Elena. Thank you very much, Elena, for being with us today and for telling us what is in the menu. The floor is yours.
Thank you very much for this introduction and all this energy. Very excited to hear all the talks, starting from Roche and what the real need from the end users and what are the solutions currently available from the photonics side. And here we have the full supply chain came together. We have some end users, we have research and development, we have people who are developing optics, spectrometers, all the elements, photonic integrated circuits, light sources who are integrating the system. We also have some market information, manufacturing, production. All of it comes together here, and we will hear from many of them, and we will find new solutions for today's problems. And
thank you very much, Elena. Elena worked really hard on bringing this community together. Thank you so much for your great work, Elena. She is our expert for life science and medical. So the EPIC members with business in medical, you have to talk to Elena. She managed to know each and one of you and what you can do in the medical market. And let me also remind you that this slide only reflects the companies who register for this meeting. So if you're missing your logo there, you're an EPIC member, we did not forget about you. We never forget about our members. You just simply did not register for your meeting. Please register for the upcoming meetings that are relevant to your business if you want to be part of the supply chain, which is only a graphical representation of the participant list. I also would like to say now hello to the people who are watching this meeting in YouTube. So hello, YouTubers. If you want to get in touch with any of the participants today, all you have to do is send me an email, jose.pozo@epic-azoc.com, and I will be more than happy to make the introduction. But of course, feel free to use the chat if you have questions real time. I will read them in the meeting room. And this is, of course, also valid for the people in the room. We have the internal Zoom room chat. You can use it to find customers and get in touch with each other. After the meeting, when the meeting is over, we are going to arrange one-on-one meeting rooms for you to actually do business. So stay. When the meeting finishes at 5 o'clock, we are not done. We continue with our meetings, with our one-on-one meeting rooms. And with this, it is time to start. As promised, we're going to hear from the end users. We're going to hear from the key pharmaceutical companies in this world. Monica is from Roche. She's here with a shopping list that we all can fulfill. Monica, thank you very much for supporting Roche. I know how difficult it is in these times to talk from the pharmaceutical industry, but we want to help you. We want to help Roche being even greater than already is. The floor and the attention of everyone, of course, is yours.
Thank you very much for this invitation. I was actually pretty glad to be able to talk today. I'm just going to share my screen, and I hope you guys can see my slides. So again, hello everybody. And thank you so much for this great pleasure of being able to talk today. I would like to talk to you about the current state on manufacturing. I work for Roche at Basel, and I work as an MSAT engineer specialized in purification, which is so-called DSP processes. So I just wanted to give you a close introduction what I actually do, because there's a lot of like abbreviation. So MSAT stands for Manufacturing Science and Technology, and this is my home where I work for, and we build the link between research and development and the manufacturing world. So while all the products and processes are being discovered and developed at the research and development department, once those processes are in the phase of being transferred to manufacturing, this is where I come into play, or colleagues, our team, we are in charge of transferring the process into the manufacturing facilities. So we perform the facility feeds, we upscale the processes in order to produce at our facilities, and we act as the process owners in order to support production in troubleshooting activities. At Basel, we have a different portfolio of molecules, going from molecular monoclonal antibodies, bispecific antibodies, and also the so-called antibody drug conjugates, which is the project that I happen to be working currently. This product, which are also called biopharmaceuticals, are produced using cell cultures. So they are produced in a liquid fermentation, and they have to be purified. And this is my area of expertise. And for the purification process of the products, we usually use a couple of steps of chromatography and also ultra diafiltration in order to formulate the drugs in the final solution. And then we transfer them into the facility in which they are filled for the final distribution. So the current state is processes and products, as I said, are discovered at the research and development departments. They usually perform process characterization in small scale models. Here, the critical attributes of a product are identified, and also it goes hand in hand. So once you define which are the critical attributes of a certain product, you of course need analytical methods to be able to detect those attributes. And here is the place, I would say, is the perfect place for new technologies to be introduced in a process, because once you are in the process of developing something new, then you are completely free of introducing new technologies there. We have currently some processes which are coming in the future, which are already implementing. I would say it's probably too old for you already, but it's new for the pharmaceutical world. So we are trying to implement Raman spectroscopy, for instance, to facilitate the analytics of some of those attributes. And we also perform, for instance, at the DSP part, we are introducing UV spectroscopy in the form of the Solo VPE, for instance, to be able to measure UV outline and saving a lot of time in the sample preparation. During the process transfer, we of course identify the gaps between the donor and the receiver side, and this is also a chance of implementing new technologies. But of course, that is not the end. It doesn't mean that we are not able to implement new technologies at the manufacturing plants, but it is of course a little bit more difficult. It is more time consuming, because then you have to take into account all the GMP regulations. And once a process has been implemented, yeah, you know, like never touch a running system. It is much harder to implement something in a running process at a production level. Here in the upstream processing part, as I said before, we perform cell culture. And these cell cultures have to be monitored, and this requires extensive sampling in order to be able to detect many of the attributes that have to be controlled, for instance, glucose, lactate, product concentration. And here there are some already projects which are trying to implement Raman in order to be able to see those attributes online. At the DSP world, we are a bit more interested in the process performance part. So we also have extensive sampling in order to perform pool analytics. So we are interested in the product concentration, but also in the purity throughout the process. Then finally, but like not least, is the analytics of the final product. So here we are, of course, we also need analytical methods to detect all the attributes that have been defined as the critical ones, and all everything that the product has to fulfill in order to be released. And then what are our future needs or challenges? So I would say the central goal of the whole pharmaceutical industry is reducing the lead time, reducing the time that is required from discovery into marketing. As Jose said, it takes sometimes more than 10 years from the discovery until going commercial. And every single person in the industry is trying to reduce this time. So we are, of course, eager to have better analytical methods that leads us to a better process control. In terms of future things that might be implemented, we could talk about process intensification and integration, going from batch to continuous manufacturing, but also the so-called personalized medicine. Here we are going from producing large masses of a product into very specific products that have to be developed and produced in a very small time. So here, as I said before, challenges for the analytical methods. They have to be, of course, compatible with our GMP regulations. They have to be scalable. There are a lot of very nice technologies out there, but they are simply not compatible with the manufacturing world because they cannot be upscalable, or they are not reliable enough. So going from the housing up to the software, whatever, like all those details have to be taken into account at the moment of thinking about implementing something in the pharmaceutical industry. And then, of course, the final part about releasing the product, we need what that's so-called real-time release. We need to decrease the time that is needed for releasing a drug. And here, of course, more sensitivity, more specificity of the analytical methods can also help. I hope I didn't go too fast. I also forgot to put my timer, so I hope this is fine. I'm open for questions right now.
Perfect, Monica. Thank you so very much for your great presentation and this very nice overview of what is already implemented there in the process monitoring. It's really great to see that you already use some photonics technologies in there, but let's see how we can help you to improve it and indeed how we can decrease the time from a development of the drug to the production. So, Jose, what do you think, how we can approach this problem best?
How we can approach the problem best? So it was very clear in the slide that we all love, the slide that we all want to keep watching and that I am going to use a lot in the future, that there is a role for both Raman and solo VP 190 to 1.1 micrometers slope spectroscopy measurements. So it is clear that we have some room for cooperation. So I would like to ask you, Monica, if you could share with us maybe what we call the Santa Claus Christmas list. You know, this kind of company profile, don't tell us the name, this kind of company profile that you would be very happy working with maybe in a couple of months.
I'm not sure I understood the question.
What would be the ideal
correctly.
partnership we could do during this meeting? You mentioned Raman, and Raman is, of course, a very interesting technology. You call it a new technology, which was for us, I'm sure, quite surprising because we've been quite couple of long decades already working on it, well, deploying it. What is currently the state of the art? Why do you call Raman a new technology? What kind of technology are you using that could be replaced by Raman? Exactly.
So what I said, it's for us it's new. It's, of course, the technology has been there quite for a long time already. For us, what's new is maybe one of those key projects that are being developed right now is the so-called sample-free bioreactor. So this is unfortunately not my area of expertise. I am more in the part of downstream processing, but for the fermentation world, for the upstream processes, for cell culture, it is very attractive to be able to monitor the cell performance online. So Raman is being implemented currently in order to measure, for instance, glucose, lactate, cell growth, but there are still some other challenges that are not being able to be measured online. And those are the things that might facilitate, like in the future, a better monitoring of the fermentation.
The floor, of course, is open for questions for everyone, but since I know in the beginning already with their first coffee they're a bit shy, then I'm going to make it easier for them. I'm going to call for volunteers within my friends. Monica, have you heard of hyperspectral imaging, basically a camera in which every pixel can measure a spectrum?
No.
You're going to hear a bit more. I have a friend here, Marc Larive from Xenics. Marc, you were smiling the whole time, so you were expecting me to give you the floor. Marc, when it comes to process monitoring for many industries, Xenics have been pushing for hyperspectral imaging being a key solution. Do you have some track record on pharmaceuticals and how this technology could be relevant, interesting for a company like Roche?
Yeah, hello everybody. Hello Monica. Yes, we are working in hyperspectral. So you see in my presentation. In fact, we in Xenics are not manufacturing hyperspectral system, but we are designing the camera, which is at the I see high-spec thing. Yeah. There are other plenty other here also doing hyperspectral. Okay, so we are just doing the camera, but we know that the camera, which is let's say implementing a system with a spectrographer, and that's what the companies such as the other that are here are doing, is a good solution to both have the information on the location, so where the thing is happening, and on the content spectroscopy. That means in each location, which is the spectroscopic signature of what you are looking at. So it gives you, and I will talk a bit about that because I know that it has been used for something and it could be used for pharma. I'm sure that.
Okay,
and
so you mentioned INDATECH, so I have to give them the floor. Thierry, good afternoon. You look awake, you look full of energies. Tell me, Thierry, what is, in your opinion, the business proposition for hyperspectral imaging in pharmaceuticals?
Yeah, actually, we have developed an hyperspectral imaging technology that we are currently promoting and developing for Raman imaging applications. So in this case, we could contribute with Raman imaging, meaning that we will not only give the Raman spectrum to identify the species, but we should also be able to give a spatial distribution or an information that where you can control the chemical spaces or active ingredients of your products. You mentioned glucose or lactate or these type of things, but in a way that we can control the distribution of a meaningful area. So my question would be that I would be very interested to discuss further with you how to use it. It will not be as fast as to be used online, but it will be fast enough to be used at line. So that's the idea about it.
Very, very good point that you raised there, Thierry, because we are going to go now to talk a bit about Raman to Thorlabs. I'm going to go to Verena first. But Monica, is a boundary condition to have full real-time process monitoring?
What do you mean if there is a boundary?
Do you do or do you need to do all the monitoring of the pharmaceutical process in real time, or is there a room for not so 100% real time?
That would be great, but that's by far not the current situation. So far we are, of course, even the things that are being monitored online are still are sampled and monitored offline, because we still do not trust completely the online systems. But that would be great, and that would be the only option to actually implement continuous processes in the future, because without the online detection, you will not be able to implement process control.
Monica, you need to stay till the end with the presentation with Julio Hernandez from High Specs, because you're going to be quite impressed on the real time of hyperspectral imaging. But before that, there's a company in EPIC that everybody knows called Thorlabs. And Thorlabs, they told me that they're working on a Raman spectrometer. And I thought this could be the perfect position to tell Monica what Thorlabs is doing to this respect. Verena is here with us. Verena, good afternoon.
Good afternoon. Hi. Thanks for inviting me.
Thank you for being here.
Yeah. So here at Thorlabs, we have established a new group which is dedicated to spectrometer development, and I might share my screen. So yes, one of our first products is the Raman spectrometer for substance identification. And this spectrometer is based on a unique technology, which is the coded aperture. And this coded aperture is used instead of a slit in the front part of the spectrometer. And here you can see, I hope you see my mouse as well. You can see the huge coded aperture. It's nearly 2 by 3 millimeters. And compared to this, a spectrometer has usually only a slit of 25 by 100 micrometer. And the advantage of this is that we can collect a lot of light while maintaining the resolution of our spectrometer. So, and as Raman is a very weak effect, it's beneficial that we can collect as much light as possible. So yeah. And another thing we have, which might be interesting for you, is that we have a very large sampling spot. So maybe it helps you when you want to analyze a bunch of cells or something like this. So it would be interesting to see how we can analyze your probes. And as you can see already, we already have a library of pharmaceutical substances, and our system is capable to analyze a mixture from up to 4 substances. So, yeah. So I would like to know from you, what is your biggest challenge when you are doing Raman spectroscopy?
So I have to say I'm not an expert, but one of the challenges that popped currently, and that came from it came actually from how to say it's I think it's been imposed as a requirement from the pharmaceutical authorities, so FDA and so forth. When you have a multi-product facility, as we are trying to implement right now, so you have a facility which produces several products at the same time, of course, different lines, you have to be able to prove that there is no cross-contamination. And because our products are so alike, it's quite difficult to have like one analytical method that will tell you like if the other products that are being produced in the manufacturing plant are there as an impurity, and especially if they are only in traces in comparison to your main product. So I would say that that's the main challenge that where Raman could definitely save us a lot of time. If like one method could actually do that instead of having to apply several orthogonal methods in order to prove that the other components are not there.
Yeah.
Great, thank you so very much. Monica, we will come many more times to you with all these different techniques we are going to discuss here today. But also during your presentation, you mentioned that in the process, you need to consider all different steps and all different parts of technology, which is of course imaging system. But as you also mentioned, there is a big component, which is the software. And here today with us, we have Andreas from Prediktera, who can tell us a lot about the software, analytical software. Andreas, would you like to share your screen now, and let's hear what software solutions are there on the offer for pharmaceutical industry?
Yes, thank you, Elena. I will share my screen here. Can you see my slides?
Yes, and
Great.
Let's go to presentation mode. Perfect.
There we
The
go.
floor is yours.
Thank you. So my name is Andreas Vidman. I'm the CEO and one of the founders of Prediktera. So what do we do? Well, we work with a technology called hyperspectral imaging that has already been mentioned a bit today. This technology really opens up a new dimension in analysis of materials. to summarize, kind of a short explanation, what this technology does is basically doing chemical analysis in an image using a camera that can see visible light or often then near infrared light. And with such a camera, you get a spectral fingerprint in every pixel of the image. And then using machine learning software, this is where we come in at Prediktera, you can turn this spectral fingerprint into information. And this technology has a lot of benefits, but it's fast and non-destructive. So therefore, it's very suitable to use inline in processes for real-time applications. And also, it's very useful for spatially heterogeneous materials, because you have this spatial domain, as this is an image actually. And so we do software for hyperspectral imaging analysis. Just to be clear, we don't make cameras, we make software. And we have the software suite to go from idea to a solution. And our goal is to make this technology, hyperspectral imaging, to make it easier and faster to implement for companies, and therefore reducing the time to market. And we've been working with this technology for quite a while. Already in 2007, we started working with this technology. So we have a lot of experience both in the software development, but also working hands-on with different applications. And based on this experience, we have developed our own software tool, which has the features that you need to go from an idea to a solution. And we worked in many different fields: food, agro, pharmaceuticals, etc. Various types of applications like a classification or quantification analysis. Could be laboratory or could be something that's used in the process. So the offer that we have for you is a software suite to go from R&D to real-time industry systems. So this could be doing kind of an exploratory analysis early in the process, so to speak, when you're looking into this technology to do R&D. We have something called Events, which is software for that. We then have another product called Breeze, which is then useful to develop a kind of a routine analysis application that you then can apply as kind of a laboratory test, or if that's a first step as a small scale test before you go to kind of put this in line in the process. But anyway, this is the Breeze software, and we developed a lot of nice features in the software, and also we put a lot of effort into making it user friendly, so it actually can be used with a touchscreen by an operator. So you don't need to be an expert in spectroscopy and so on to use this. Software then to train your algorithms, to train your applications or develop your applications. And then finally, we have this software called Breeze Runtime, which is a prediction engine basically for high-speed data processing, to take whatever you have done in Breeze and deploy it as a real-time or embedded application, for example, inline in a process like a sorting application or a quality monitoring application using hyperspectral imaging. And can recommend also, I assume everyone gets these presentations afterwards. There is a link here to our website. You can go in and see a little video of one of the applications we have been doing with the food industry. So what we are looking for, I would love to talk to you to discuss your applications that you might have, and maybe you're interested in doing some feasibility studies to test your application or some of your ideas to what can be done with hyperspectral imaging. So we're looking for customers, partners. This can be kind of end users, process industries who's going to use this in their own processes, but it could also be integrators and machine builders who want to implement this technology in their systems. So please contact me. This is my email, and please visit our website as well. There's a lot of useful information there.
We will. Thank you very much, Andreas, for a great presentation. And we have a lot of questions for you. The first one is coming from a company in Belgium. The company is called Caleste. They make fantastic imaging sensors. And the company is represented by Jan Vermeeren in the YouTube channel. And he's asking you, how is your software coupled with the camera? Is it with a Genicam or a dedicated API?
So we support cameras. It's a very good question, first of all. So thank you for that. So we have a couple of, or I would say 3 or 4 suppliers that we support, specific camera suppliers that we support that can communicate with our software. And we use typically their SDK. So that's typically the camera suppliers has an SDK that we can integrate into our software. So this is typically how it's done.
Okay, so you are very flexible to any camera manufacturer that needs to have their own software. That's very clear. Is your software suited for spatial as well as for spectral scanning?
Yes.
Okay, so this is tell me.
I mean, if it's I assume the question is relating to the kind of, because there's line scanning cameras, and then you have maybe spectral scanning cameras and so on. So it's all hyperspectral, but it's just how the data is acquired. And I would say yes, you can apply our software to any type of hyperspectral data. Yeah.
Okay, so let's ask the potential integrators of that software into their cameras to see how they can use it. So I already introduced Julio, who's going to be your last speaker, but I'm going to now ask him from the buyer's perspective. Julio, HiSpecs has made a paradigm shift in the reduction of the size and increasing the dynamic bandwidth. When it comes to the firmware, I know that you spend a lot of money developing the software yourself. When you saw the presentation from Prediktera, is there any room for potential cooperation, collaboration? Andreas is smiling because most likely you're already working together.
Yes, we are.
Yeah, well, we have a very close collaboration with Prediktera. We've been working on that for quite some time now. We are actually offering currently all our systems with a free trial version from Breeze system, from Breeze software. So that's also, I mean, for us as camera manufacturers, it's a really great tool because it allows the end user to start working from day one, right? They can start doing image acquisition, start doing modeling, doing analysis, generating interesting results. Because as you know, we've been working with Breeze for quite some time, and they are very user-friendly. They have Andreas and the team have.
I'm gonna go back to Monica, Julio. And the reason why I'm going to go back to Monica is because I want to ask you something very important, which is, Monica, when it comes to cooperations with the supply chain, does Roche develop their own instruments, their own systems, or what kind of companies regularly supply instruments, equipment, machinery to Roche? Are there more instrument manufacturers or system manufacturers? We want to identify the supply chain for you.
Well, no, we do not build our own equipment. Unless there is something that we need like super customized, and in which case we usually collaborate with the different companies out there to build on a specific solution. But I would say from my experience, the suppliers for analytical instruments that are like currently being used, like especially for UV spectroscopy, we are working right now with the solo VPE technology. So we are trying to implement this throughout the network to facilitate UV measurements and saving time in sample preparation. But we are not married to a company. You know, if there is something out there that allows us to improve, then of course I think Roche is more than open.
Indeed, let's clarify this. They are looking for R&D cooperation. So any company that you think you have a technology that can be suited for the pharmaceutical industry, they can help. Do you know that there is something called supercontinuum generation that can actually illuminate a very long range of wavelengths? It's a company in Denmark, NKT Photonics. Do we have anybody from NKT Photonics in the room? I think we do. He's
right up here.
Hello. So what do you think we could explain, Monica? What is the value of using perhaps supercontinuum generation or illuminating a long range of wavelengths for the pharmaceutical industry?
Right. Maybe start with just a lot of people may not be familiar with what exactly that word means. So a supercontinuum laser, as you refer to, is imagine a laser type light source. But instead of emitting one wavelength, it emits every wavelength, just like a lamp, just like any other broadband light source, from say 400 nanometers in the close to the ultraviolet up to well into the mid-infrared. So that's a kind of light source. Recently, we've seen a lot of interest from the pharmaceutical. In fact, that's why I'm participating today, to try to find out exactly why these types of light source lend themselves so well to various types of hyperspectral sorting applications.
You will hear later with even system integrators like Thermo Fisher on how we can actually work together. Before that, we go back to theory. We go to Lambda X. Monica, listen to this carefully because it's going to change the life as you know it. Thank you very much, Thierry, for being with us this afternoon. The floor and the attention of everyone goes to... no, sorry, I made a mistake. Sorry. We go to Xenics. We go to Marc Larive. We go to Xenics. Sorry, Marc. You mentioned Lambda X before, and then I got confused. Marc, thank you very much for being with us today. Tell us how your detectors can enable pharmaceutical inspection. The floor is yours.
Okay. Hello, everybody. Thank you very much, Jose. Let me share my screen. You see something? It's okay?
It's just maybe let's go in presentation mode.
Yeah, that's good.
Yes, perfect.
Okay, so hello everybody. My name is Marc Larive and I am working in Xenics as a strategic marketing manager. So first some word about Xenics. Xenics is a manufacturer of infrared solution. That means we are deeply involved in infrared from short wave to long wave, and including mid wave. So as you can see, basically from the visible to the long wave. We manufacture sensors, cores, and camera. Especially in short wave, we master all the technology, so we can provide just the sensor or the core or the camera. And we propose either linear, either 2D system at sensor or camera level. And for example, we have a very long line 2K pixel with very high speed, 256 kilohertz for the full line per line. And for the 2D, the fastest one we have is 1.7 kilohertz in VGA format, full format. So that's basically what we are doing. Again, we are doing the camera, which is included in more complex system, but which is the core of the system. Some word about the hyperspectral and multispectral. Okay, I think a lot of people already talk about that. Typically, with a 2D camera or sensor, you use one direction to get the spectral signature and the other direction to get the location, in fact, of what you are looking. And then using typically a push broom system, you can have a global online system analyzing both what you are looking at or where you are looking at and the content of what you are looking at, because you can make the spectroscopy of the items. So you can do that with a 2D array. You can also do that, with a 1D array, big line, and then in this case, instead of getting grating in front of it, you multiplex the LED lighting in front of it. So again, you select a specific waveband and you have the let's say, the information on this waveband on each pixel, and then you change the waveband and again. So it's let's say it's not hyperspectral, it's not as fine in terms of information as an hyperspectral, but it's probably sometimes a more cost-effective system that you can build, and always based on an infrared cameras. And for us, we have specific shortwave cameras that you can use for that. So typically in pharma process, what the shortwave infrared can help you, where the shortwave infrared can help you, and what is it for? I would divide it in three. First, you can see inside with shortwave. So that means once it's in package, and even if the package is not transparent to the visible, that means you cannot see anything with your eyes or with a standard color camera. If you use a shortwave, then you will see inside. You will see the level of the moisture of the liquid inside, and then you can say whether the package, the bottle is properly filled or not. You can also detect some leaks like this. It works also on, let's say, pills inside blisters. You can see whether whatever the blister is transparent or not. You can see and even analyze what is inside the blister to check that you have the pills effectively really inside or not. So typically, this is done using an infrared camera, but you have a bigger system doing this. This one is made by AT. So that's the first line, the first pass. The second pass is to see what you are looking, and to be able, using the HSI, hyperspectral imaging, to check what is the content. So again, you use typically a shortwave camera, and using the spectroscopy on each pixel of it, and you do that real time, you can analyze what is the real content of what you are looking at. Here, for example, in visible, you have all white pills, but if you look and make an analysis in hyperspectral, then you can see that there are two different pills. Okay, and again here you have 4 different pills, and you can make the difference between the aspirin and vitamin and so on. So again, this is done at a higher level, but basically, or typically here by hair wall, here in resonance, by resonance, but it's basically it's made of shortwave imager, and in front of it you put a spectral analyzer, and then you make a processing so you can really say what you are looking at. And the third pass, again using hyperspectral, you can see what you are looking, but also how much you are looking, how much there is. So here, again, it's based on a Xenics shortwave camera, and you can make the analysis, spectroscopy, and then you can see how much quantity you have and how much, let's say, real active content you have in the pill, and where is it located in the pill. So you can do that on the pill, but you can do that on let's say, a large distribution of pill. So you can check the concentration, whether you do the right amount, the right concentration of active content, but you also can check the homogeneity of it. Because if you want to take half a pill, the content has to be homogeneous. Or if you have to take 2 pills or 1 pill, if one is nearly empty and the second has twice the content, then it's also an issue. So that's really a good way to check the, let's say, the quality of the process for the pharma processing. So in terms of what we are looking and what we can provide, so let's say that looks at the next step and check what are the opportunities and how to identify the next steps. At infrared camera levels, what we need the, let's say, some information from the user regarding their need regarding techniques, but also regarding the business, so that we can optimize. We can optimize either in the wavelengths, because the camera has a limited wavelength, typically 0.9 micrometer up to 1.7 micrometer, whether you need to go further or not. Optimize in terms of resolution, typically for hyperspectral, how many spectral information do you need? So that means how many lines. Optimize in terms of form factor. Usually the infrared cameras in shortwave, they are line scan with just one line that you scan, or 2D in a 4x3 standard. And this is due to the, let's say, the movies and the standard vision. But as now we are not just doing standard vision, but you want something smarter. Let's say you can perhaps need, I don't know, 10x3 form factor or something else, depends. So this typically we can discuss about that. And then also optimize the camera at Xenics regarding what you need. And typically could be, okay, in my system, I don't need all the array, but just part of it. Or I don't care whether there are some dead pixel, but providing that it's in this direction, so we can make specific bindings or this kind of thing. Whether you want to embed it some filters, I already spoke about that in a previous presentation, to do multispectral, but with a kind of smart camera. So this has to be done in partnership with the customers to know what wavelength they need. And finally, to put more intelligence on the sensor on or the camera, but which kind of intelligence? Be sure that Xenics is not competing with its customers, so we will not do the full hyperspectral system, but perhaps we can put some intelligence that will simplify the work of the hyperspectral manufacturer and give a more cost-effective system at the end. So that's all, let's say, the paths that we are investigating, and we need some partnership with people, typically people that are in the audience. So thank you very much, and if you have any question, I'm open for discussion.
Marc, thank you so much for this presentation and on this great view of what are the capabilities from hyperspectral imaging for pharma industry. Before we get to your very interesting list of requirements and looking for partnership, I would like to take this opportunity, as I'm speaking already, to have a chance to ask some of my questions in there, because you showed in your presentation that with shortwave infrared, we can look inside of the containers. We can look whether liquid is there, whether the pill is there. We can also identify what pills we're looking at, or perhaps also what liquids we are looking at. Can we do it all at once? Is it possible to see inside of the package that pill is in there and what pill you have in there? That's also in process of sorting, or maybe like the composition of the pill. So to look past the packaging and through the distribution of the active elements in there.
Yeah, in theory, it's let's say you will do spectroscopy. So providing that you know what is the content of the blister or the package, you will have the result hyperspectral. So you just remove the part that you know is there, and then you get the rest in principle. I mean, but again, at Xenics, what we are doing is just the camera. To be very clear, we are doing the camera, the best camera that we can offer to the equipment, and then all the trick in hyperspectral, it's not us. We give the raw, let's say the raw information, pre-processed information.
Okay, good to know and very clear. Thanks for clarifying once again for me and people in the room. So let's see what we got in the room, how we can help you, what we can integrate in there, and what the hyperspectral camera manufacturers think about it. So let me see whom we addressed first with this interesting question, or if anybody has any questions already in the room. Okay, people are being shy, that's fine. But we do have high specs here with us. So once again, Julio, the floor can go to you. Let's see what can you tell Marc how he can help you further in improving the cameras.
Sure thing. Well, hello, Marc, again.
Hello, you.
High specs and Xenics, we have collaborated in the past, right? So for many years, one of our main sensors, the sphere cameras, were using Xenics sensors, right? So it's a partnership that we had in the past and that we would happily reignite it. However, what we're doing today, as Marc knows, is we're using MCT detectors for our sphere sensors. And the spectral range we're using for our MCT detectors are from 1,000 to 2,500, which last time we discussed this, we're a bit outside of your strategic development line, if I'm correct. But I mean, the collaboration with Xenics and other sensor manufacturers is basically, it boils down to spectral range and spectral resolution. And of course, readout noise, which is critical for having highly accurate measurements. But yeah, well, this is more like a chat we have had several times with Xenics in the past. But well, I think Marc and the rest of the manufacturers are aware of our requirements and needs. That said, we've been working with Xenics in the past, and they are an excellent company, and we would gladly work with them again, right?
Great. So thank you very much. Yes, indeed, that's what one of the paths we are investigating to extend the wavelengths. For the moment, let's say standard in gas is limited at 1.7, and that's why I understand that you are currently using the MCT. What we are looking at is getting an extended wavelengths further to 1.7 with a proper noise, and more than that, with, let's say, how can you say that, an efficient way of using it without moving parts, something compact. And okay, so because MCT, I'm coming from MCT, so I know what are the requirements for it. So it's also a bit complex. So either this is a solution, either there are solutions. Currently, we're investigating on how to offer more than the standard 1.7. But I would be very pleased to talk to you and to anybody who would be interested in it to check and to really, let's say, fine-tune the requirement to make the best choice so that we can provide you with the best, let's say, the most efficient solution.
For all that will be great. Yeah, so I guess we can take this conversation on the side anyway.
Okay. Good. Thank you very much.
Thank you.
For the users in the room, you should know one thing. We already talked a little about Raman. We talk a lot about hyperspectral, like in every meeting. Hyperspectral has a huge boom. Monica, also the other users, do you know that there's a technology, terahertz, that goes up and down? I call it the roller coaster of photonics. Terahertz is booming again. There's a couple of companies who are developing terahertz sources that are optimized for process monitoring. We have one of these companies here in the room, Lytid. Anna Golinelli is here with us. Anna,
yes.
You join a meeting of pharmaceuticals, but you make terahertz sources. What business opportunity do you see here?
Yes, so the terahertz, yeah, as you say, it's booming. Hopefully next year will be booming even more than now. And yeah, if I can share my presentation, I'd be glad to show you what we do. Okay. Can you see it?
Yes, crystal clear.
Perfect. I'm going to presenter mode. Okay, so this is, so thank you again. I'm representing Lytid today, and Lytid is providing terahertz solutions. So what is terahertz? Terahertz is part of the electromagnetic spectrum that stands in between microwave and infrared. And for this, it has particular and exceptional features, like, for example, it can see through plastic, foams, and non-metallic material, but it also sensitive to water content. So for this, it is highly suitable for non-destructive imaging. So in the specific market of pharmaceutical, what we can propose is, for example, chain quality inspection in the production. For example, we can see through, as you can see in the image here in the bottom, we can see two tablets in a blister pack, and we can see whether there is a tablet or not. It's like something that we can do. It's similar to what Xenics, what Marc shown, see inside. So as you can see also in the tube, where the tube is transparent, we can see through the tube. So what we propose is a new method, because the terahertz is becoming now popular. It's taking some markets in the non-destructive imaging. The image that I'm showing you are real time, so they are really fast. They are non-destructive, as opposed to X-ray. They are non-invasive, and we can obtain sub-millimeter resolution. So I am presenting now this technology that can be used in the pharmaceutical, and what we are looking for is some feedback from the pharmaceutical companies. So we'd like to know whether, we'd like to know some feasibility study. We'd like to know whether this new method for seeing through packaging can be interesting. We'd like to know also the performance that are required, whether a real time is something that is important, or whether resolution is more important than real time. Everything in order to improve our method. So on.
Thank you so much. You know, one thing with terahertz, I'm a huge fan. So we were gonna be putting it at every football stadium, almost every room, to detect weapons. People realized that if it was a rainy day and you were wearing a wet coat, they couldn't see the weapon inside your coat. So it was not that efficient for security. However, when it comes to spectroscopy, when it comes to actually determination of contents in solids, this is gold. The question from Monica was, can we make this a suitable real-time operation? Do you have some examples on how this could be used in real-time determination of pills, as my colleague Elena said, inside the package?
Yes, I'm not an expert in pills. When I was talking with Elena, through the recruiting process, I told her, like, we were talking about a lot of applications that are used for pills, but unfortunately that's not my expertise, I have to say. But I do think that there might be also applications for the liquid pharmaceutical drugs, as for instance, when a drug is filled into a vial, you of course have to control that all the vials are filled. So this, I can imagine it can be used there, like, quite easily, also for what Marc presented before. It's super interesting. I have to say I have no clue how this controlling is being done so far, because I am also quite far away from the packaging colleagues. So I'm super sorry. I am so useless.
No. You've been gold. That's why I'm coming to you all the time. No, don't worry. You're our entry point to Roche today. Let me go back to Anna for a sec. Anna, you have a clear roadmap to set up this kind of technology for process monitoring. Which companies from this room can help you get in there? What's your preferred future, and which companies will help you get there?
So it's something. So we provide sources, and we provide the detector, but we do not provide cameras. So if any of the present would, we are open, of course, to always check new terahertz camera technologies. For example, we know that there are terahertz camera technologies on the market, but we are always looking to new solutions.
Okay, no, see, you're looking for a terahertz camera. You look, you look on the right place. So I would like to say hello from TeraSense. I know they are watching. Welcome to Epic whenever you want. But here there is an opportunity for cooperating. And Monica, you want to hear about Raman? You pay attention to the next presentation because this guy is something very special. INDATECH is with us in the room. Thank you, Fabien, Chauchard-Rios, for being with us this afternoon. Tell us about Raman for spectral monitoring in pharma. The floor and the attention of everyone goes to you.
Okay, thank you. So yeah, in INDATECH, we are a bit a bridge between sensor and user. Maybe I can share the screen, but you will see my presentation also focus on hyperspectral imaging. So I will just share it. Wait.
It's coming. It's almost here. Here we are.
Okay.
Yes.
So just switch to the full. Okay. So you should get. So I will just maybe introduce quickly INDATECH. So it's a company we formed in 2009, mainly based in what we call process analytical technology, which is mainly the application of optical and spectroscopy technique to pharmaceutics and mainly process. So we have been bought by a bigger company a few years ago in order to expand our business. So as you say, we work with Raman spectroscopy mainly for biotech, but we also develop many other sensors because in some cases you have to use UV spectroscopy in line, and other case you have to use hyperspectral imaging. So we have some kind of approach quite wide in order to focus to the different topics in pharma, biotech, and chemicals. And I will say there's many need, and I think we saw that with the presentation from Roche that maybe you don't need to go inline in order to provide something new. You just have to help to go faster in the process of developing a new drug. And you can even do that by proposing innovation even at the lab or process dev, what we call the development of the process. And what I would like to propose is maybe an example. And I think what is interesting is that in this key example, there's several presenters that we already saw. So I would like to introduce an example where we were working with one of the biggest supplier in vaccine and biotech. And he was looking to analyze better what some kind of their biotech product based on hyperspectral imaging. So we had to go quite fast for the development of this new product that we call HyperReal. So we rely on several technology leaders in Europe. So the first one was Specim. So we work with them in order to use their hyperspectral camera, which is a FX17 with a range from 900 to 1700 nanometer. And we also work with Prediktera. I think you probably can see the graph on the top. And we have been able to develop, based on these different technologies, something that make hyperspectral imaging as easy to use as using a photocopier. Because the idea was to have something very fast to use in the lab by technicians who don't know hyperspectral, and in order to have quick information about the drug that is being developed. So everything is included in some kind of drawer, and then the machine make automatically the measurement. So I will give you more detail on this need. So in fact, when you make vaccine, and I think with the current vaccine that you hear about the COVID, you can see that it's critical to manage the storage. Currently, the vaccine, sometimes you have to use very low temperature to store them, but another way to stabilize the protein or thing like that is to use what you call freeze drying, which means that you will convert the liquid into ice, and then you will remove completely the water with a specific process, and then you will obtain some kind of solid product that you can store easier than a standard molecule. But something is critical, that you have to be sure that there's no more moisture remaining in the cake, what they call the final product. Otherwise, you cannot store properly the vaccine. And it's also important to validate that the solid is not broken, some kind of cake. And I would say the physical aspect of the product is also important to be able to dissolve quite faster. So we designed some kind of plate where you can put directly the sample that are into glass vial, and we're able to make the measurement through this vial and we're able to measure directly the moisture. So you can see completely on the right, a customized report that we have done by working with Prediktera. So the user have, I would say in 1 or 2 minutes, all the value of the different lyocake. And I think they want really lot of time, because making a standard measurement in the lab with what they call a Carl Fisher for so much sample can take sometimes 3 weeks. So when you try to develop a new drug, a new vaccine, and to see how to stabilize it, it's critical to go fast in this step. And by using this optical technology here, the hyper spectral imaging, you can go much faster in the development time. And it's also very important for the clinical trial to validate the dose that you are going to test. So on this approach, I would say it's a real breakthrough, and I think it's a good example of what you can do with existing technology on the market. Maybe I will just switch what we are looking at. So as you can see, we are really quite focused on end user. I mean, we work in many companies. We work with biotech company in Korea, US, and in Europe. So we are really eager to work further with this kind of company and to develop some application. It can be in, as I show you, in hyperspectral, but also in Raman or other, because we have many knowledge about how to implement this technology in a robust way in order to gain faster time, but also to gain in safety. We have also worked with some other company, not necessarily pharma, but more company that want to analyze through their bag or through their capsule, some kind of powder. So this is the end user, but we also work with OEM, and I can provide some examples that we have developed technique to measure directly inline the lyocake with one leader in manufacturing lyocake. I mean, it's a GEA. They make a lot of machine for making lyocake. We work also with Pharmatech or Proditech, which are supplier for vision machine for tablet analysis. So now we propose them NIR solution, and they are able to analyze completely the production. So these are the main topics, and we also work with distributor, and currently we are also looking for some collaboration in the country I just listed there. So we're also happy to work with this kind of people.
Thank you very much. This is what we call the epic question. You know, what can you do for them? What can they do for you? So you're looking for a distributor in England, Ireland, Germany, Greece, and Portugal. Is that correct?
Yeah, we already have a quite good other distributor, but in this country, yeah, we are still looking for some people, yeah.
All right, so I would like to call here our network. We do have many potential collaborators on that, and we'll get back to you in a separate note afterwards. But I would like to ask you now on the technology perspective. You identified yourself, you said both I'm an end user and a camera spectrometer, spectral imaging solution provider. I'm going to ask you now from your perspective as an end user, what would be the big challenges that the end users are having to incorporate hyperspectral imaging in their systems?
I think hyperspectral imaging currently is quite mature, because you can see you have a lot of sensor. Here, for example, camera from Xenics or other supplier. I think if you look at the size of the hyperspectral camera, now they are already much more smaller. So before it was something 3 or 4 times bigger. So now you can integrate it much more easier. And the computer also work much more faster. So you are able to have an analysis of the hyperspectral image quite fast, because an image is very heavy, and I would say 10 years ago it was critical to analyze the image in fast way. So these are the important points. But something important when you work in pharma is mainly the environment. You have to take care of what they call the GMP rules in order to not contaminate the drug. But even when you make a software, you have to be compliant with many rules, like the Part 11 and other. And this is a very big work. So it's more or less where we work, because of course INDATECH is not providing sensor. We are using sensor, and we see all this work that is necessary to work on the software and on the integration and how to make this system compliant. So I would say that with the pharma you need many actors, and I think that's why the solutions are quite expensive currently.
Fabien, for me, what we need is to incorporate more and more the semicon manufacturers, the volume production companies. This is what we are missing. And we have in the room Sumitomo Electric, we have in the room ST Microelectronics. Actually, I'm going to go to ST Microelectronics. Oriana from ST Microelectronics, good afternoon.
Good afternoon.
Thank you very much for joining today. It means a lot. ST is a very engaged member of EPIC. What do you look for in this market, and what kind of cooperations would you be looking to start after this meeting?
Thank you for the question. Actually, let me say so far, very interesting. Very interesting. I like the approach. I hope I can say that.
Of course.
Because there are different perspectives. There are the end user perspective, which is really important. And this is one reason why I'm here today, because we are looking at, in general, healthcare, but with particular focus on pharma, which is very complex, highly regulated. So there is on both sides the needs, the interest to understand the needs, but also, especially on the manufacturing side, how we can really match these, because we experience these medical device companies very complex. You say that also, Monica, and I understand we're entered along the manufacturing process, the entire development process. Even if I'm wondering, even in Spectral is a fantastic technology and is already existing technology, as Fabien said, which is great, because something is developing something new, something is what we have. On the other side, how this existing technology can match also this cycle that where anything is GMP compliant, everything is more, under control, scare me a little bit, but it's, I'm here to listen, to understand how all these end users see, first of all, the type of technology, and then we will think about that and try to see if we can help. That's for sure.
Thank you very much. I want to say that ST has been very engaged on both sides. Also, as an end user for process monitoring on the semicon processing, we had a separate call on that. And today you're looking for understanding what is the need for sensors in the pharma industry. So thanks a lot, Oriana. I'm going to go now to a company who is, by definition, a success story of photonics, Thermo Fisher Scientific. Thank you very much, Igor Nederlof, for being with us this afternoon from Thermo Fisher, a company that we all love and want to have more engaged. The floor and the attention of everyone, I cannot stop saying it, goes to Thermo Fisher. The floor is yours.
Thank you for the enthusiasm on this. What's a great afternoon in the Netherlands. I will try and share my slides, I hope. You see them? I will go full screen.
Almost there.
Almost there.
But,
yes, that there?
Here it is. Yes, fantastic.
Yes, so Thermo Fisher is a very large company, and we have over 75,000 employees now, 5,000 of which are R&D scientists and engineers. We invest 1 billion in research, and we have a revenue of over 25 billion. Yeah, our mission is to enable the customers to make the world healthier, cleaner, and safer. I do want to have a side note before I start with my slides. So I'm from the electron microscopy division. So what some of you may know as what used to be FEI, before that, Philips Electron Optics. So my slide will be how we're engaging in pharma with this electron microscopy business. And I think in the academic sphere, we really saw cryo-EM take off. And cryo-EM is the imaging of biomolecules, so proteins, protein complexes. And in the last 10 years, it's gone from a method that used to be called blobology, where you see blobs on the left, to a method now where you can actually see your atoms, and it's more of a chemistry technique. So in 2017, the Nobel Prize was handed out for this technique. And basically now also pharma is taking note. And pharma is buying electron microscopes to indeed look at molecules, look at biomolecules, but also to see now how small molecules bind to these molecules. So this is mainly done, let's say, in the research atmosphere. So in the discovery. And how cryo-EM works, I have a movie. I was brave. I hope it works. So this shows you how the technique. So your protein is soluble and it gets frozen, and then it goes into a microscope where you do your imaging. And so you image your proteins and you take many images. So you see them here, and you collect the different angles of your proteins. And once you shoot more and more images, you can do, so you assign angles to them. And then if you have enough of them, you can solve the 3D structure of your protein. And so you can look at the chemistry, you can see how drugs bind, maybe how, for instance, an antibody and the conjugate binds to that antibody. So once you've solved the structure, you can use that to design new drugs. So like I said, that was mostly done in the discovery, but we're now also moving more into a GMP environment where we're looking at, for instance, particle size distribution of samples of, for instance, adenoviruses that are laced with RNA. You can look at the purity of your samples, the integrity, also the morphology. For RNA, you can also see if the RNA has entered the viral particles or the liposomes that you're using as drug delivery vesicles. And you can also do cell bank testing. So this is now, all of this is working in a GMP environment, meaning that they have the regulations that are needed for QA and QC to bring these drug molecules to the market in the end. So what does Thermo Fisher do to help people adopt cryo-EM, since this is a relatively new technique? Of course, we'd be happy to sell you microscopes. Since we're now part of Thermo Fisher, we also have leasing options through Thermo Fisher Financial Service. But we're also working with a global network of CROs and companies that want to, let's say, try out the technique and see if it's right for them. So we have special starter kits, explore and impact packages to get them up and running. And yeah, if you're interested, I'd advise you to come visit our website or contact me later.
Thank you so very much, Dankjewel, Igor, for this insight into Thermo Fisher's work in pharmaceutical industry. I'm sure that there can be a lot of cooperation in this room as well, so don't be shy. People, do ask your questions. We are here to do that, to collaborate. In the meantime, I will start my question, since you mentioned that with cryo-EM, you can also look into the viral particles, and you're talking about it in terms of also of drug development. Is there any contribution you also are running now, if you can talk about it, with the current virus pandemic going on?
Yeah, so I didn't get into the application that specifically, but I think it's very interesting that you mentioned this. So I think the virus was first identified in November of last year in the genetics were solved in February of this year, and in March already the spike protein, so the important protein that's sitting outside of the virus, was identified, and that information indeed is being used to develop a vaccine and develop drugs. So it's very exciting that cryo-EM also took a role in that development.
Igor, goedemiddag. That was really great. I think it's the first ever presentation from Thermo Fisher in an online technology meeting, so you have the honor. I want to help you sell microscopes, of course, anytime. So what I'm going to do is I'm going to go to the Confocal. I'm going to see how they can get your microscope in something relevant to Roche. Can we do that? Is that a way that we can try to find this kind of cooperation? Saneke. It's a very Dutch meeting today, but of course Dutch people rule. So you have great tulips and great windmills and great Dencon focal. Sanneke.
Yes, hi Jose. Yeah, thanks for having us here. And I see lots of, think friends from earlier projects here in this meeting as well. And maybe I can share just a few slides to show the people that do not know Dencon focal yet what we do. Because I do think there are possibilities for collaboration.
With Dencon has been great. So far, we made so many introductions as system integrator.
Yeah.
Today, Dencon focal. Let's see how we can help you.
Yes. So let me make it full screen. Yes, here it is. So unlike some of the people I've heard present today, we are not really focused on one specific technology. So we do not sell, for instance, just hyperspectral images, for instance. But we, as a system integrator, we do the design engineering and sometimes also assembly of specialized optomechatronic systems. And we are active in various fields, and we're trying also to get into the analytical field and into pharmaceuticals, and to see where we could have a role in that. We are based in the Netherlands, and mainly our customers are OEM companies. So we usually develop a new system based on a technology idea. We develop the full system and make it ready for production. And we do that with a lot of people in-house with knowledge and expertise in different areas. And our core knowledge or our key differentiator is the combination of optics and vision with mechatronics. But as you can also see, we also have the supporting expertise in-house as well. For instance, mechanical, electrical software. And there's a large focus on quality engineering in our company. So we're both ISO 9001, but more importantly, ISO 13485 certified. So we also know how to develop according to...
and you recently opened your office in Singapore, and we are already working very hard at connecting you with all our members in there. Dencon is one example of cooperation with EPIC. Thank you so much. Allow me to go back to Igor. Igor, today we heard in the beginning from Monica from Roche about the need to do most of the work real time. Thermo Fisher is looking for some collaborations to bring technologies into real-time processing, because most of the technology presented today were not really for real-time.
Yeah, so in terms of cryo-EM and looking at, let's say, the atoms of these biomolecular structures, it's very difficult because they need to, the electron microscopy is in vacuum. So what happens is the samples get frozen before they get into frozen before they get into a microscope. So doing this real time, it would be a challenge. But I think we are looking into this as well. Yeah. And there are, for instance, smaller companies that make flow cells and things like this. But yeah, we don't do that ourselves, but there are companies that are picking that up. Yeah.
Thank you so much, Igor, for being with us. I'm going to come back to you during the next Q&A, and we're going to find ways to collaborate, because as I said before, for us, Thermo Fisher is a company that we really, really want to help from the European photonic industry perspective. Let's go to Amsterdam Scientific Instruments. Jen-Liha Bilal Kalyonsu is here with us to find opportunities for collaborating. Let's go to collaborate. Let's find operationals with Thermo Fisher, with Dencon, with Roche. The floor and the attention of everyone goes to Jen-Liha, goes to you.
Hi, hello, good afternoon. Thank you very much for the opportunity. I'm going to start with sharing my screen. So
it's here, it's coming. Yes, perfect.
Perfect. And let me do it full screen also for you. So I guess it works.
Yes, perfectly.
Cool. So I am Bilal from Amsterdam Scientific Instruments. Obviously, we are based in Amsterdam, and we make hybrid pixel detectors for various applications, pharma processes, biological samples, etc. And let me explain you what kind of stuff we do. So I'm gonna start with our product range. We make hybrid pixel detectors for electron microscopy, for X-rays, And we also have a unique product called TimePix3Cam, which allows you to like detect ions, neutrons, photons in the optical range in any kind of device when it's combined with a microchannel plate or a phosphorus plate. And we are also open to do OEM products if any of you are interested in the special features that I'm going to explain in the following slides. So I'll start with a very brief explanation of this technology, if this term is kind of new to you. So this is the picture of the sensor. It's invented and still being developed in CERN. So it's made up of 2 layers. At the top, there's the sensitive element where a particle interacts with and creates a signal. And at the bottom, there's the pulse processing element. So what is special here is that actually each pixel, both of these layers are pixelated, and each pixel is like an individual detector. It has its own comparator, it has its own readout channel, etc. So what you see here is actually the real microelectronics picture of one pixel. So it's full of electronics. And what is most important thing here is that the high signal to noise ratio, because these pixels here allow you to set energy thresholds. Depending on your application, you can set them as you like, but if you set this threshold, the lowest threshold right above your noise level, then you're gonna eliminate all the noise at the detector level, not during the post-processing, but at the detection. So this allows very high signal to noise ratio, which is very useful for low dose applications, for beam sensitive samples. Like if you are trying to image in life science applications for some biological molecules, proteins, pharmaceuticals, they are affected by the electrons or the X-rays that you send on them. So you have to use low doses, otherwise you're gonna get an image of a destroyed sample. So this technology allows you to use short exposure times or low doses. There are other features as well that I don't have time to explain much, but it also allows very high speed acquisition with 2000 frames per second. It allows continuous acquisition with 0 dead time in between frames. And with the TimePix3 chip, it also allows very, very fine time resolution if you're interested in the timing of events. So to tell you a little bit more about the applications, I want to go through one by one with the products by explaining one example from each product. So that's an electron microscopy detector, which we mount on an electron microscope. So here is a comparison image. If you do a diffraction experiment, that's the kind of a picture that you would get. If you're not familiar with it, that's also fine. But I want to show here, these 2 images are taken with 2 different cameras, CCD and the Cheetah camera, and with the same position on the sample and the same amount of dose. On the Cheetah image, you see here lots of weak diffraction spots, so that you get much more detailed information about your structure compared to a conventional CCD or conventional CMOS camera. So you get more into your, you get to know more of your structure. And this allows you to get structures of some molecules that were not possible to image before. One example is the hen egg white lysozyme. So the structure of it is solved first time with the Cheetah camera. And another example is the orthocytomol with the hydrogen bond bonding them. And the author says this is the first pharma having an unknown structure solved with the ab initio 3D electron diffraction. So that's one example with the electrons. And the other example I want to give with the X-ray detector, which is our Linux series. So here you see spectral imaging. You see overlapping foils of 4 different metals. And so this is a colored X-ray image, because I mentioned this energy thresholding in the first slide, but you can also set multiple thresholds. And if you assign these energy ranges with different RGB values, for example, in the end, you can get a colored image where you can differentiate with different materials in between just by taking simultaneous images at different energy ranges. And the last one is TimePix3Cam. That's a very unique device. As I said, it detects the optical, or detects in the optical range, but can be combined with different microchannel plates that are sensitive to ions or neutrons, etc. And this is special in time of, if you are interested in the timing of particles that arrive on the sensor. For example, at the top right, this is an example from a quantum communication experiment where they image 2 a pair of entangled photons at distant locations because it's so sensitive with the timing, and it also senses like single photons, single electrons, etc. And at the bottom, there is the mass spectrometry imaging of some enzyme where the time of flight is used to get information. So it's also very useful for in situ measurements. So this brings me to the end of my talk. So I try to give you a summary of the technology, products, and like possible applications with this technology. Thank you very much.
Thank you so very much, Bilal, for this very great overview of capabilities and offerings of Amsterdam Scientific Instruments. I would like to come to you first with what we call epic question. It goes as what you can do for them and what they can do for you. So you already presented on your technology and application. So you basically did give some answer to the first part of the question. But when it comes to the second half, what they can do for you, we have a full supply chain of photonics industry here, who is also interested in pharma applications. So what is there you're looking for?
Yes. So for the photonics part, especially the Timepics 3cam, which is this device here, is quite special. So the sensor there is called Timepics 3, and it has this unique feature of timing resolution and also other features with single electron sensitivity. The data acquisition is also different. The readout mechanism is different. It takes a lot of time to explain all this, but what the point I want to come to is that we are open for generating applications with this technology. So this technology is new. It's very well known in electron microscopy industry. The name which is usually used there is direct electron detection. For X-ray, it's photon counting detectors. But for the Timepics 3cam, it's very new. And we know that this device is pregnant to many different exotic applications, especially when it comes to time of flight with this sensitivity. So I would like to invite everyone to look at our brochure and the full specs of this device. And if they see a potential use of this, of any of these features of this camera, we'll be happy to get in contact, collaborate, and generate different applications that the uniqueness of this product can be used in to serve science, basically.
Yeah, sure, absolutely. And same as me, and I'm sure many other people in the room, Mike from Photonics Insights is quite impressed with this camera. And Mike, I would like to ask you to maybe give your thoughts and comments on it, and also maybe in meantime to comment on what is the present and preferred future for Photonics Insights, and how companies in the room could help you.
Well, thank you very much. I mean, we're really focused on the low end, meaning low cost spectroscopy, right? So seeing some of these instruments here is quite inspirational because we immediately think of how can we do it for less cost, right, and broadly distributed. So that's the first thing, and that's what we're looking for. Is we're looking for partners in that realm. So one company we're very interested in. I'm sorry, I just have to look at the name again because there's so many names. It's a Prediktera. Love to speak with Prediktera. We have some data that we can share with you that could use your analysis. One. Two, Bilal from Amsterdam Scientific Instruments. I can't wait to look at your, at your brochure because I'm sure we have multiple use cases that we could share with you where you could use your tool. It was an impressive presentation. So where we are, we develop low-cost spectrographic instruments, and what we're looking for is we're looking for partners to collaborate with in order to develop use cases. We already have an active use case in Sri Lanka where our low-cost spectrometers are being used to currently characterize the tea harvest. So if anyone is interested in partnering within that realm, food process technology, we're highly interested in collaboration. And thank you so much for the mention. We really appreciate it.
Fantastic, thank you. But if we go back to pharma, maybe we can come once again back to Monica and ask you. So you see now there are different technologies, they come with different offerings, but now we also have low-cost spectroscopy. Is that really would that be really an issue, or something could be giving a preference to a technology, the price of that technology?
I don't think the cost is the limiting factor for the pharma industry. I think we're interested. It sounds like I'm investing from my own wallet. No, but I'm like, beside jokes, I think what makes the pharma development so expensive is the tremendous amount of time that is needed. Like as Jose said at the beginning, from discovery and all the regulations that we have to pass to be able to bring one product into the market. So I think the cost of the instruments is not really something that would motivate us to implement something. I think it's more the technology that allows us to be faster, to be better. So I think if we might improve our processes by being able to save time, then the investment is very much welcome.
Fantastic. That's fantastic news, I think, here. So we don't need to worry about the price of the technology, just to make it best and to improve the process. And with that, I want to address to the final presentation of this meeting. Back to hyperspectral imaging, back to the company which we mentioned so many times because it is so great. So let's hear what Julio will present to us from HiSpecs today. If you would share your screen now and your presentation, we would love to hear it all.
You see my screen, right? Yeah.
Yes. Well, very clear. Thank you.
Good afternoon, everybody, and thanks for staying here for more than an hour and a half. That's quite nice of all of you. Well, yeah, I'm gonna talk a little bit about what we do here in HiSpecs and how we think we can actually contribute to the pharmaceutical processes, production processes, and research processes both. My name is, I say, Julio Hernandez. I'm the hyperspectral applications manager in HiSpecs. I have I'm a physicist by training. I've been here for over a decade, mostly in R&D. Now I am working with new applications for our instruments. Right. One of those things we're interested in exploring is pharma. So who are we? HiSpecs is part of a larger company called Norsk Electro-Optic. Norsk Electro-Optic is a optoelectronics company based in Norway. We have been in the optoelectronics business for over 35 years now. We were formed from as part of the defense research establishment in Norway. And our developments in hyperspectral imaging have been ongoing for over 50 years. It started as a part of European international projects, and it became a commercial success as well for us, right? So we have different instrumentation that for different applications. Our technology development driver is our instrumentation, which we call the HiSpecs Classic, which is mostly oriented for scientific and high-end industrial applications. We also work a lot in remote sensing. And also recently, well, not recently, a couple of years ago, we started a new line of products for industrial applications, right? And by industrial, we mean applications in which spectroscopy can bring in information that is not easily available by traditional machine vision systems. We work with food industry, pharma industry, recycling industry. We work with a lot with precision farming lately, which is quite an interesting subject. We work with water management. Anyway, spectroscopy comes in all colors, right? So we can work with many different applications. And where do we see ourselves working with the pharma? Well, first of all, why are these processes? Why are these technologies relevant for pharma processes? Well, we think it's because spectroscopy is a proven technology that can contribute to understand better how pharmaceutical products and processes are taken care of. By adding the imaging capability, then we can add an additional dimension for space which basically tells spatial properties of those spectral properties that we're seeing. If we contrast this to more traditional point spectrometry, in which we get an integrated signal from an object that we're illuminating, or from which we are receiving some light signal. In this case of spectral imaging, we can also get contextual information from a peel, or from a rock, or from a leaf, or from a fruit, or from a building, depending on the scale that we're looking at. So basically, it gives us spatial information that we compare to the very highly detailed spectral information. How can this be used in general? In very general terms, it can be used to classify, identify, and quantify spectral properties of the different objects. In pharmaceutics specifically, classification can help us to identify the good from the bad thing, right? So what is what you want to have in your components, and what is what you want to exclude. Identification goes to the next step. It is basically what specific material or component or API is present in your or specific contaminant for this cross-contamination that was mentioned earlier. Which specific materials are there, right? So with high spectroscopy resolution, it's possible to identify with high accuracy what's in an image. And finally, quantification. Quantification is important for example, if you want to. Some pills, they are supposed to be delivered in halves. So it's important that each half contains the same concentration, right, to be effective. So in this case, it's important that to have an adequate monitoring system that will tell you whether or not what you're looking, what you are producing, complies with the quality parameters. So I would say this: adding hyperspectral imaging technologies to our process will enhance the capabilities of seeing beyond the traditional image vision technologies. We have been discussing about the importance of real-time measurements, real-time results, inline and online results. So hyperspectral sensors in general are capable of doing all these things, right? So we can implement our sensors or other sensors on online processes that will be able to provide real-time results. We work very close with Prediktera for our own analysis and to provide solutions to our end users. This is a very quick example of the kind of thing that we do here in our labs. This was part of a project in which we were trying to do some analysis for a feasibility study, in which, our customer sent us some samples that they wanted us to identify and classify, and then to work the mixtures for different components. So we got the samples, we imaged them with two of our cameras, with our classic, which are our workforce instruments. And then we created some spectral libraries that characterize in detail each of those types of materials that they sent us. And then we work with the mixtures, right? So in this plot at the end, in the chart at the end, basically what we're seeing is how the different mixtures are composed. You see the last two columns, mix 1 and mix 2. They have different weight values for the three different excipients plus the API. So in a very visual way, we are able to retrieve information about the peel or the powder before compression. We were this was quite a long experiment. We were able to determine some changes associated mostly only to pressure changes for the compressing rates. The spatial distribution for us is basically a given. We will be able to tell with high accuracy what is the distribution of the different materials in a scene. We have a quite high spatial resolution, so we think that we can provide this type of information in a reliable, repeatable, and timely way. Right? Our technology, in general terms, we think that the high-spec instruments are a very robust solution when it comes to data quality. Data quality both from the spatial and the spectral points of view. Spectral, we have high fidelity, high repetibility, and stable calibrations. Spatial, we have low aberration and similar thing, thermally stable and easy to deploy in different configurations to image different type of scenes and size of scenes. We work in the 400 to 2500 nanometer area. We have three main sections: the veneer, the sphere, and the intermediate sphere, which covers to 1.7 microns, similar to Xenics was explaining earlier. So depending on the application that the customer is interested in evaluating, we would recommend one or other or a combination of implementation. Like I said at the beginning, our main driver for technology development has been our classic instrument for scientific applications, but we also have the industrial version of it, ready for deployment, ready for scaling up, deployability, and we have our own SDK. We use third-party sensors. In the past, for example, we use Xenics. That allows to have a smooth implementation on site, on a production line, on a research lab. It's very flexible technology. Right? And that's it. I'm sorry if I rush in. I hope you.
Thank you very much for a great presentation. HiSpecs is a great company. We already love you so much. We even had a virtual company visit when you had your product launch. Everything is in YouTube. So if you want to hear more about this, we went virtually to visit the company for the release of their latest camera. Today you came to an EPIC meeting, and you know what EPIC is all about. It's about cooperation. So I'm going to ask you the EPIC question. We already heard a lot about the companies in the room. What can you do for them, and what can they do for you?
Well, what we can do, for starters, we would gladly help you out to evaluate whether or not hyperspectral is what you need for your processes, right? Many problems can be solved with point spectrometry. Many others can be solved with multispectral imagers. Many can be solved by other types of spectroscopic techniques or optical techniques. Some of them can be the challenging ones. We think that hyperspectral can definitely contribute. When will this be mostly relevant? When spatial information at the small scale is relevant, right? Spectral fidelity is quite important, but it will depend a lot on the process. We can also help to develop custom solutions in terms of inline integration of the system, sensor development to some extent. We can help out to modeling development for specific applications. That's the kind of thing we can offer.
Thank you very much, Julio. And we're going to make a lot of introductions, more than we already made. There's some people who want to talk to you. I know Dieter from Photonics is also very impressed with your technology. Dieter, good afternoon. Today you see some potential cooperation with Julio or with any of the participants in the meeting. Please tell us in a couple of sentences what Photonics does.
Hi, Jose. Wonderful meeting. Thank you.
Thank you.
Photonics comes from a grading business, and we manufacture spectrometers that with the transmission optics, so they're very sensitive. So any application in spectroscopy that needs a lot of that sees very little light, like Raman spectroscopy, is the prime application. So I was all ears all day in this meeting. But there's also like the hyperspectral cameras that also need a grading, and the transmission gratings can be manufactured to be extremely efficient at specific wavelength ranges. So there's also some cooperation. And then we also need sensors to detect the light. And there was this talk about from Amsterdam scientific instruments about these wonderful sensors with like no noise and super sensitive. And so this is all fantastic.
I was also very impressed with Amsterdam Scientific Instruments. I really want to know more about the sensors. I'm a quantum physicist, so whenever they tell me about the sensors with low noise for photon counting, is fantastic. I'm going to go to another company member of EPIC, Limo. Because Dirk, you joined this meeting. Limo is a great company in the micro-optics revolution that we live in Europe. You joined this meeting looking for potential collaborations. We heard for 2 hours of your life potential collaborations already. Is there anything that you can do for others or others can do for you?
Yeah, thank you, Jose. It's a really fantastic afternoon, and I I got the really impression that the applications are really at the beginning, and there are so fantastic possibilities in case of using, for example, photonics and photons in the right way. And our task, for example, could be that we can control intensity of photons on surfaces using our beam shaping technologies, and we can do it in a volume or even in a very well-defined area, so that we can illuminate with laser sources, with lamps, with from UV to infrared, with well-defined intensity distribution and well-defined profiles. And that's something we think could be interesting for analysis of materials. And therefore, this could be something that I can introduce into this meeting here.
You know, one other company is very interesting, Dirk, is Opto. Marcus is here. Marcus is here in the room. We know Opto in the microscopy market. One company that is collaborating with many EPIC members in this field. We heard a little bit about Pharma. It is difficult to get the end users in the room, but they are in the back office here into this presentation. What learnings do you have from this, and what is your preferred future for Opto going even further in the pharma industry?
Yeah, thank you for coming back to me. So no, it was really exciting hearing all these aspects here. So yeah, I found very interesting having the sensors, having the beam splitters, or having the filters for the hyperspectral. As you said, we are more in the microscope field, but we see a lot of dynamic at this moment in flow cytometry, in the microfluidic applications to build in microscopes. So, and when I speak about microscopes, compact this size, and here we were speaking about fluorescence, fish technologies behind, and I saw a lot of photonics and research going on over the years, and now chips are available, microfluidic chips that are doing very special instrumentations possible, and laboratories. And we are the guys to make it work inside machineries, and that's why the people are coming to us to feed the robots with it, and we are doing the imaging therefore. We are a big cancer research and all these technologies. So Roche searching for automation for high-speed imaging or detection, and having all these technologies. No, yeah, I know most of the people here. But okay, we are small with 50 people, so we are just at the beginning, but the big OEMs are on the doorstep. So to use all these technologies, yeah.
Thank you very much, Marcus. You've been excellent in following up with introductions. One more for you. There's a company called Enlir that makes fantastic detectors. Peter Totrov, thank you very much for joining today. 2 hours of your life talking to the supply chain, hearing from the supply chain. What can you do for them? What can they do for you?
Thank you. Peter Totrov, I'm the CEO of the company. We are doing spectroscopy for the mid-infrared. We do a very fast, unique combination for 2 to 5 micrometers. So we are kind of the extension of the visual, the near-infrared, and then the extended. So what you can do here is to find some OEMs or system integrators that can use our technology into the pharmaceutical. We have experience already with the pharmaceuticals. We have done several fermentation and bioreactor projects, and so we are already in there.
Thank you so much for being with us. You are really raising a lot of interest in this industry already for the last 5 years since we know each other. Congratulations on what you're doing on this field of detection. Monica from Roche, I want you to close the meeting. Well, I will say a few words after you, but I want you to close the meeting. You came here looking for potential collaborations and friends. We have a lot of people who want to be your friend, Monica. What do you take away?
First, I have to thank you for the invitation. It was a great meeting. It was great hearing so many applications that might have a potential application. Actually, I am I'm thrilled by all these meetings, all these talks that happened
we
today.
made Roche thrilled. That's as much as we can do, Monica. This is the first one of many meetings that we want to have you in the room, because I just want to say that in a meeting like this, in 2 hours, we actually spotted cooperations in Raman spectroscopy for the raw materials, in hyperspectral imaging for the pills composition, in terahertz for looking through the package, and even we were all amazed. Amsterdam Scientific Instrument, we are all amazed with your detectors. I want you here. I want you here in this flyer. Thank you very much, all of you, for being part of this. Once again, I just want to say that we do all this to help you. From North Pike, next to the sea, in the Netherlands. Sometimes the sun is shining, sometimes it's not. It's freaking cold right now. Thank you very much for being part of this amazing adventure of connecting 641 companies. We keep growing. On behalf of a fantastic super team of photonics experts, 15 people, half of them with PhDs, I just want to say that we're going to continue. We're going to continue organizing these online technology meetings, and you already have the calendar for all the meetings in Q1, and that includes forensics, ultra-fast lasers, even photonic integrated circuits. I just want to say keep registering, keep giving us feedback, because we want to get better, and most important, keep being epic. Soon, in April, I'm gonna start traveling again. I'm gonna travel like crazy. I used to travel 200 days a year. My family is not gonna see me for 2 years, because I want to see each and every one of you. From North Side next to the sea, until the very next time, take care of each other, wash your hands, and stay healthy. Bye-bye.